Hello, everybody, and thanks for coming to listen to this talk, which is titled Bitter Nectar, Toxic Pollen, Pollinators, and Plant Chemicals. For the next half an hour or so, I'm going to talk to you about the somewhat surprising fact that when bees and butterflies and flies and wasps and other pollinators go to flowers to get their food, they sometimes encounter very toxic chemicals made by the plant. And we'll talk a little bit about the consequences of this, how the animals handle it, what the outcomes are for pollination and for the plant. Yeah, and so I will try to remember to tell you the names of the bugs that you see in some of these photos. This is a Morrison bumblebee feeding on a milkweed flower in Nevada. It happens to be a pretty toxic diet, that milkweed. So this is something of an outline for the talk. I'm first going to talk about some general background stuff about the plants that have these chemicals, what these chemicals are, and some of the hypotheses trying to explain why plants would do this, why plants would have evolved to put these potentially toxic chemicals in the attractant for their pollinators. Um, uh, and then I'm going to talk a little bit about some of my own research that I did. It's actually 10 years ago, but it's from dissertation research. And I'll be looking at some of the benefits to pollinating bees. I'm going to talk about pollinators, which includes a vast array of vertebrate and invertebrate animals. And I will mostly be thinking about bees when I do this because that's my specialty. But just to state it here, we need to remember that these consumers of floral products are diverse. There are flies like this one that I was lucky enough to see foraging on a flower in the Aleutian Islands this summer, to butterflies of all sorts, and moths and wasps, ants, and of course bees. When I say bees, remember you can generalize that a little bit to pollinators as well. So where are these toxic chemicals found? Well, it turns out that a giant array of different types of plants produce toxins and put those toxins into the nectar and the pollen of flowers. We're used to, you probably are familiar with the idea that plants defend their foliage and other plant parts from herbivores or chewing animals or sucking animals by putting toxins into their leaves and such. And so here's the surprising fact that they are also putting those chemicals into the flowers. And I've just rounded up some of the ones that I'm familiar with or interested in here. And I'll go from the top center photo and go clockwise and just explain them. The wasp is on a tilia flower, a linden flower. We have some bees visiting a milkweed to the right there. And then going down, we see catalpa. We see rhododendron, California buckeye in the middle at the bottom, camas, iris. We have a squash or a pumpkin flower. And then we have astragalus and solanum right in the middle there. This is just a tiny subset of the plants that produce these chemicals in nectar and pollen. So just be aware that this is not a rare thing in nature. And it's actually pretty extensive in the diet of flower foraging insects. Um, and so what are some of these chemicals? Well, I've listed a few here based on their chemical classes, like alkaloids and terpenoids. The alkaloids that you may be familiar with include caffeine from coffee and tea, but it also occurs in citrus flowers. Nicotine from tobacco, toxins. Actually, the photo and other imagery on this slide is about these toxins, which are chemicals made by rhododendron flowers. Not all rhododendrons, not all azaleas, but some of them. This is a chemical that human beings have been interacting with for a long time because honeybees will collect the nectar from these plants, make honey out of it, and then when people gather that honey and eat it, they experience a range of psychotropic and unpleasant effects, including paralysis if you eat enough of it. And my little example here includes this coin that shows King Mithridates, I think, a Turkish king from 65 BC who actually use these chemicals in warfare. And this is not the only example we have of people using plant chemicals in warfare mediated by chemistry of the flowers. So in this case, it's said that he left honey on the battlefield, and the enemy, the Romans, went and ate that honey. The honey was tainted with toxins. The people became sick and then were easy to take care of. Just be aware that human beings interact with many of these toxins on purpose, but also sometimes not. So others in this list include thymol, which is that minty smell we get from thyme, and oregano. It's found in a range of plants, including the nectar of that linden tree that I showed before. Glycosides like uabane, which is, excuse me, just one second. Uabane is a cardiac glycoside that comes from milkweeds. It is an extremely toxic chemical and is commonly found in milkweed nectar. Phenolics like gallic acid, which are known to be healthful for people and often turn up in nectar. The saponin example is called esculin, and this is this really gnarly chemical that comes from buckeye trees and is absolutely toxic to some insects that forage on it. In fact, it's a commonly observed thing that you'll find dead honeybees at the base of some of these trees. So they are foraging for the nectar and they are dying before they can even leave the tree. By contrast, there are some other consumers like bumblebees that seem to be, and carpenter bees that seem to be fine with consuming esculin. So more on that in a minute. Ammonia, non-protein amino acids, and just so many other chemicals. And I'll just quickly say about non-protein amino acids. These are structurally, they are amino acids, but they're different from the ones that you and I use as the building blocks for proteins in our bodies. They have been found in virtually every nectar that's been sampled for them, maybe every nectar that's been sampled for them. So they're ubiquitous in plants. They're not necessarily toxic, but they can have some impacts on consumers. Speaking of consumers, before I go on with this slide, let's remember that bees and butterflies and other things that forage at flowers are consuming plant products or plant parts. And there's really no distinction between doing that and feeding on the leaves. So they are herbivores, just like a caterpillar that's foraging on the leaves of a plant. In fact, this plant to the right here in the photo, you can see that it's been chomped on by foliar herbivores that are caterpillars. And it's also a favored nectar and pollen source for some bees and other things. And so let's remember that these flower feeders are herbivores. Let's also remember that not every flower feeder is a pollinator. So a pollinator is an insect that takes that moves pollen from the male parts of one flower to the female parts of the same flower or another flower, allowing the plant to reproduce, right? This is a benefit to the plant, but the pollinators are not, with a few tiny exceptions in nature, they are not doing this out of altruism or on purpose, if you will. They are foraging for food. So there are many flower foragers who are not pollinators. They are thieves or robbers, or there are various other pejorative terms in the literature of science to describe them. But let's just keep in mind that when I say pollinator, that means something that actually helps the plant reproduce. And we really just mean things that forage on the flowers, right? So we should say flower feeders, maybe. Back to this slide, why would a plant such as this turtle head poison its mutualists? There have been observations of this kind of thing for about a century, and people have come up with an array of potential hypotheses to explain it, which I've summarized some of here. So one would be just to protect the floral resources, the nectar and pollen. And here we might think of protecting it from flower visitors who are not pollinators. Nectar robbers are the classic example. These insects, mainly bees, that will tear a hole in the flower near the base, and they'll suck out the nectar through that hole rather than going to the front entrance of the flower and entering, contacting the plant sexual parts and taking pollen away with them. But there are lots of other types of non-pollinating foragers, and maybe some of them are kept away from flower resources by really nasty chemistry. There is some evidence that this is true in some systems. Maybe these things are antibiotic. They kill microbes in the nectar and let it and keep it from spoiling. There is evidence of this as well. Maybe they manipulate pollinator behavior in some way. For example, there is a non-crazy hypothesis that some of these chemicals make pollinators drunk or inebriated in some way, such that they handle the flowers differently and they're sloppier and they may deposit or pick up more pollen. There is research that's demonstrated that some of these chemicals make the pollinators fly a little faster and work a little quicker on the flowers. There are some behavioral things that may be changed by these compounds. Another hypothesis for why they would be in nectar and pollen is what we call pleiotropy. This is the idea that the plant is making these chemicals somewhere else in its body. So for example, in this case, it's making these two chemicals called aucubin and catalpol to protect its leaves from foliar herbivore caterpillars, but the plant just can't help it. And the chemicals go everywhere in the plant's body or the plant, including in the nectar and pollen. I'll tell you, for the most part, we've ruled that one out. We know that plants can restrict chemicals of all sorts to different parts of their flowers. And of course, it's possible that these chemicals attract pollinators, and that is where I'm going to go with this talk. We could talk for days about all of the negative outcomes for pollinators or plant flower foragers of consuming these things. But I want to talk some about the ways that these chemicals can benefit pollinators, and this is mainly because that's what I've done my work on. The last thing to say about this slide here is just to remember that we're talking about plants. Plants are rooted to the ground. They cannot walk around and find mates. They cannot run away from their predators or their herbivores. And one of the ways that plants get around this lack of mobility is through chemistry. And so I think you're going to see, as I talk about this stuff, that some of these things are elegant solutions to the problem of being rooted to the ground. All right, so now we know a little bit about where these chemicals occur and what they're called. Um, and what are some of the theories for why they occur? So let's talk about how herbivores, including pollinators, deal with these toxins. So these are hypotheses. Most of them, all of them, I guess, have some evidence in the scientific literature. So avoidance is one thing. The bees and butterflies could simply avoid going to plants that are toxic, and indeed we know that that happens with some things. Others may be able to tolerate some amount of these chemicals. The bees on the milkweed in the center photo, that milkweed produces a good deal of that ouabain and other toxic cardiac glycosides, and yet certain bumblebees really like it. And so it's been hypothesized that they don't like it because it's good for them in some way. They're just able to tolerate some of these extremely toxic cardiac glycoside compounds. So I took a look at data from the California Bumblebee Atlas, which is the project that I lead at Xerces. It's been going since 2022, and we have more than 20,000 independent observations of bees in our database submitted by volunteers who work with us. So this is a wonderful project. It's part of the broader Bumblebee Atlas project, and I encourage you to get involved if you're interested, no matter what state you may be in. We are active in 20 states now. But if you look at this graph, I've colored the various bars according to the subgenus that the bumblebee occurs in. So the vertical line there, you see all these italicized names. Those are the names of the bees. And then the color shows their relatedness to each other, basically. So the sage color at the top, those three are closely related to each other. The orange, all of those guys are close, they're each other's closest relatives within the genus Bombus. And there's a pattern, right? We see that the three at the top seem to be consuming a lot of nectar from these toxic plants, potentially toxic plants, whereas the other ones seem to be maybe just eating a little bit of it or even avoiding it. And the x-axis here is the percent of milkweeds in the diet, but I've adjusted it for the frequency of the bee in the data set. So if the bee was rare in the data set, that would tend to increase the estimate. If it was really common in the data set, that would downweight it a little. So we're just weighting by commonness. So I hope you'll see from this graph that there's this strong pattern where bees are eating different things, even generalist bees, and it's possible that some of these guys can tolerate these chemicals. Another really fascinating thing that herbivores do is sequestering these chemicals. Now, I can't think of a pollinator that does this as an adult feeding on nectar and pollen, but it's really common with foliar herbivores, like the monarch butterfly larva here, which consumes parts of milkweed plants with those toxic cardiac glycosides, and they sequester them in the body. They store them away from most of their tissues in a way that keeps them from interacting with metabolizing the chemicals. This is a defense against their own parasites and predators. So the parasites don't want to put their eggs on that caterpillar if it's really nasty in chemistry. So sequestration is really common in nature, and particularly I'm gonna hazard that particularly that happens with butterflies and moths. They may have ways of just metabolizing and quickly excreting these chemicals, and then it's possible that some of these things are immune to these chemicals, probably immunity acquired through long periods of coevolution with those plants. So is it possible that these herbivores could actually benefit from these chemicals? Well, I've just basically told you that yes, they could possibly. Sequestration, again, for these foliar herbivores, they're certainly benefiting from those chemicals because the chemicals make them toxic to their predators and parasites. It's worth knowing, actually, that there are trade-offs even for, in that particular example, where the animal is intentionally eating milkweed and sequestering those chemicals, concentrating them something like 30 to 40 times the strength of the way that they occur in the plant. But there are negative impacts on that animal also. So consuming these things can have benefits even when they have some detriments for the consumers. These herbivores, these pollinators, could benefit from the taste or smell of these chemicals. That could be something of an honest signal that there is a reward in the flower. So without landing, they may know there's nectar because they can smell it. And of course, these things may work as antimicrobials within the body of the pollinator or in the nest or hive for social species. And we have some evidence of that. And then finally, could these things be medicinal for bees or butterflies or other consumers? Could they help them get well when they consume this stuff? And this is more data from the California Bumblebee Atlas. This is on the vertical axis. This is the species names for all of the milkweeds that we saw Crotch's bumblebee foraging on over the last 3 years. 2025 data is not included yet. And I've broken it out by sex because there is some pattern in this particular species. They have different preferences for milkweeds, which is kind of interesting. I've circled the one at the top there, Ariocarpa. That's the one in the photo with the two crotchless bumblebees. This one is loaded with these toxins. The one below it, Fascicularis, which is also commonly eaten by the bees, is devoid of them. So it is healthy, safe nectar. But look at how much of that other one they're consuming, even when they have choices. And I'm not here to say that they definitely benefit from milkweed. We actually know that they probably don't, and there is a cost that they pay tolerating this stuff. But I think it's worth just noting that it's possible that there are benefits that we have not yet discovered in consuming this particular nasty stuff. So let's talk about the study system that I worked in for this research that is going to be the subject of the rest of this talk. So these are going to be some examples of how we do science to find out how do these chemicals impact consumers. So first of all, I did graduate work at Dartmouth College, and that was under the supervision of Dr. Becky Irwin, who's now at North Carolina State University, and in concert with Dr. Lynn Adler at UMass Amherst, and then many other collaborators at all of those institutions. And so we have two parties here. We have Bombus impatiens, or the common eastern bumblebee, and we have this gut parasite that infects it. It's called Crithidia bombi. I will call it Crithidia. It's really fascinating. It's related to malaria. So it's not bloodborne like malaria is for humans. It's gut-borne. And the way they acquire it is through shared use of flowers. So if a sick bee goes to that sunflower there and forages, she may leave some saliva or poop, and then the next bee that comes in may acquire the disease through just using the same flower and being exposed to these things. It is exactly like how you got the common cold last winter. Shared use of doorknobs and being in the presence of people who have the disease. And so the question that we were trying to ask is, could these nectar chemicals affect the parasite infection of the bee? Could it help them in some way as they deal with this significant, sometimes lethal, but often not lethal parasite? And so let me introduce you to some of the chemicals that we studied. So there were 8 of them across these 6 plant examples that I found in the literature, and we decided we could buy the chemicals from a commercial source and we could do experiments with them. So going from top left clockwise, we have an almond flower, and that's amygdalin, a really toxic cyanogenic glycoside that is found in the nectar and pollen. Next, we have buckwheat, and that's gallic acid, which I told you is healthful for people. Next, we have tobacco. The nectar contains nicotine as well as anabasine, which is even more toxic to insects and other living things than is nicotine. Next, we have citrus, and that's caffeine there. Caffeine's widely distributed in the flowers of citrus and also tea and coffee, and some other places too. Center bottom, that's turtle head, or Chelone glabra, and it produces aucubin and catapol, which are iridoid glycosides, and we'll hear much more about that plant later. And so this plant is in the figwort or the plantain family. So that includes, if you're on the West Coast, you may know monkey flower or Kekiella. That's a close relative of this guy that has those chemicals. Penstemons have them. If you've ever taken valerian, the herbal medicine for sleeping, the bitterness of that medicine is these chemicals. So there is a human use for them too. Incidentally, the bitterness of olives, if you like olives, it is other iridoid glycosides that you're eating. And then finally, we have thymol, which is found in a lot of different nectars of mint family plants and some other places as well. All right, here's the lab experiment that we did to try to find out, do these things help bees cope with a parasite infection? So we had commercial bumblebee colonies in the lab, and every day new bees are coming out of the pupa or cocoons. On the first day that they come out, we grab them and put them into this little vial here that's fitted with a nectar feeder, and there's a little wick so they can drink that nectar all day long, and we give them pollen. Every day we change out the containers so they're clean. So for 2 days, we just feed them straight 30% sucrose and a little pollen. On day 3, every bee gets parasitized by hand. So I made a solution of the parasite and I counted the number of cells of parasites in there, and then I pipetted it into a single drop of solution that they then consume. So we knew all of them started with an infection. Then we divide them in half. Half of them got a control diet, which is more of just the 30% sucrose. And then the others got one of 8 different chemicals that I've just introduced you to. And we fed them that way for 7 days. And then we sacrificed the bees and we count their parasite load. And then we also kept track of survival over the course of the experiment. I'll show you the results on a figure that looks like this. We use standard methods. It's called linear mixed modeling, but it's sort of like one-way ANOVA, if you're familiar with that sort of statistical test. We analyzed each chemical separately, but I'm going to show you the effect size for all of them in one graph. So 0 would mean no effect, and negative numbers mean that it decreased the parasite load in the bee, good for the bee. And positive numbers would mean that it increased the parasite load in the bee, so not so good for the bee. And here's the data. We find that all 8 chemicals reduced to some extent the mean number of parasite cells in the gut of the bee. Half of those, that was a statistically significant effect, and some of them were quite a strong effect. Overall, there's a no question significant decline in the bees' parasite loads when they consume any of these, or when they consume these chemicals taken all together. So this is exciting. It shows that at least in the lab, a bee can partially be healed by consuming these chemicals, but we know nothing of whether out in the field they try to consume them? Do they avoid them? We're force feeding them these things, so we really don't understand the ecological context of the bee encountering and consuming these things. Um, we were interested in costs and benefits in this experiment, and in fact, we didn't see any differential survival for any of the 8 compounds. I'm just showing all of them together here. This is a survival plot with days across the horizontal line and percent survival across the vertical. So bees died here and there through the experiment, but there was really no difference between the 2 groups. There was some mortality in another experiment suggesting that one of these chemicals could make bees sick. And this goes to that statement I made earlier about the possibility that there are trade-offs for consumers who eat these things. So we know that these secondary metabolite chemicals or plant toxins can reduce bee parasites. And I need to just stop and say I did not define this term secondary metabolites. Um, People divide plant chemicals into primary and secondary metabolites. And classically, scientists would say that the primary are those chemicals that plants need for growth and reproduction. And the secondary are these weird chemicals that we don't know why they make them. They're probably not necessary. They make fruits colorful and things, but they're not really that important. I hope this talk will convince you that so-called secondary metabolites can be very necessary to plants. We still call them plant secondary metabolites or just secondary metabolites. But here I mean those plant toxins. Sorry I didn't explain that earlier. So we have this lab experiment result. Now we want to know, are there trade-offs we can see in the field for bees that consume these things? We want to know, what are the benefits for plants? Why would plants do this? There must be something in it for them. or maybe not. And then could the bees be self-medicating, meaning that could bees be making context dependent decisions about what to eat, in the context being their parasite status and the quality of that nectar, maybe the quantity of chemicals in that nectar. So here's the field system. We're still with this plant called turtlehead. This is a wetland plant found in eastern North America. Remember, it's related to penstemon and monkey flower and a bunch of other cool plants. It has these chemicals called iridoid glycosides, and we already knew that these chemicals were found in all plant parts that had been studied, and flowers had not yet. And we know that they mediate the interactions between the plant and one of its most significant herbivores, which is this checker spot, the Baltimore checker spot butterfly. The larvae consume the chemicals, they sequester them, as I described earlier, and that helps them to keep from being parasitized by a wide array of different wasp parasitoids. So we already knew that this system was in place. And so we wanted to ask, well, is the stuff in the nectar and pollen also? And if so, how does it affect consumers who are, instead of being foliar herbivores, they are nectar herbivores or pollen herbivores? So the first thing to do was to do a couple of seasons of research on the biology of the plant, especially the reproductive biology of the plant. So I collected information about who visits the flowers. I studied the mating system, so we understand how the plant behaves so as to become pollinated and make seeds. And so in the upper right-hand corner, you can see that the flowers have, these are cutaways of the flowers. They have a male and a female phase. They last 3 to 5 days, and they're male for the first day and a half or so, where the stigma has not grown out all the way, and they're letting out pollen. After the pollen's gone, the stigma part arcs around, and you can see that in the second one. And now it is ready to receive pollen grains from a bee that enters there looking for nectar. Um, in the sort of science that I did here, we found out, if you look at the three graphs from left to right, we found out that bees do indeed remove pollen from the flowers. You can see an unvisited flower had significantly more pollen than after one bumblebee visit. There was this little tiny bee called Hyleus, which is the black and yellow waspy thing there, that would visit the flowers frequently and eat the pollen, take pollen away through sonication of the flowers, but it didn't take enough to even be noticeable through my statistical analysis there. So in the middle, we found that the plants need pollinators. They can self-pollinate or they can outcross to other individual plants, but they must have a bee visitor or some visitor who will move pollen back and forth. And then the last thing over here shows that the plants are not making as many seeds as they could in nature. So if I add pollen to a flower and I do nothing else, I let other bees visit it and stuff, those flowers make more seeds than if I just take a flower out of nature and count its seeds that it makes. So that means that the plants could use more visitors, right? They're visitor limited. The last thing to say here is that this is one of these fascinating ecological stories where the bee visitors are sonicating the flowers. They're buzzing them to get the pollen out. The pollen is hidden up above that hairy stuff you can see in the cutaways. And the bees have to vibrate the flower to get the pollen out and then pick it all up and put it into their storage areas and take off with it. So that's part of the story. And for me to gather pollen, you can see the electric toothbrush there. And that's the method. You can use a tuning fork, but an electric toothbrush is a little bit easier. So we wanted to study the chemistry of the plant also, obviously. So we collected nectar, which you see in the bottom left-hand photo there in these little micropipettes, and pollen. You can see both plant materials in those little epi tubes there. We collected all the other plant parts, and then we analyzed them using gas chromatography in the lab of Dean Bowers at Colorado State University. And she's a part of this work also. And so just in a nutshell, what we found is that both of those chemicals are present in all the different plant parts, but that nectar is doing it differently. So nectar and pollen have a little bit less than the leaves do, but the ratios are different. So the nectar occupant and nectar catalpole are not correlated with the concentrations in any other plant part. Something different is happening in nectar. So much for that idea that I called pleiotropy, right? The plants are manufacturing this stuff, and they're apportioning it in different concentrations to different plant parts. So that sets us up to know that this could be a system that's the result of some coevolution, and plants are doing something, quote unquote, on purpose. They are behaving, I'll say. Okay, and then the last part of the system is that the bees are foraging on the plants. The bees are the pollinators, but they are attacked by a number of different antagonists, and we're going to study these two. So one is our friend Crithidia that you already met. The second is this fly called Physocephala. This is in the family Canopidae. They're amazingly cool animals, and they are bee hunters. They're parasitoids of bumblebees and some other bees, but mainly bumblebees. You can see in the photo on the right that a female is waiting. She's sort of stalking that bee. They'll grab the bee very quickly. In less than a second, they will insert an egg into the abdomen between two overlapping scales of the top of the abdomen. And then the bee has about 2 weeks to live. By day 10, nothing's wrong for a few days as the larva grows in the abdomen. By day 10, the bee starts to misbehave a little, doesn't forage as much, flies funny. By day 14, they usually are, their brains are taken over by the parasitoid. They go to ground, they dig a hole, they bury themselves alive, then they die, and the fly pupates inside of the bee and then spends the winter underground like that in a safe little storage container, the dead bee, and comes out in the springtime to do it again. So we're going to ask questions about whether the chemistry of this nectar and pollen could help the bees deal with these two antagonists. So what I did is design this array experiment in one of the wetlands where I worked on the plant. There were actually 7 sites that went into the nectar chemistry work there, but this is my favorite site where I decided to do this experiment. So I took 24 stems each morning. I put them in florist water picks and then wired them to bamboo stakes in this standardized grid. And then I used a micropipetter, this little tool on the left, to add nectar solutions to the existing nectar that would result in 3 different treatments. Either the nectar was especially sweet and not bitter, so it was 1 standard deviation below the average nectar cattle pollen occupant, or it was average, or it was 1 standard deviation above the mean. So this is within the range that bees would actually experience these things in the population. As they fly through, they're experiencing sweeter flowers, more bitter flowers, and they have choices to make if they can taste that difference, right? So I just kind of hacked that system by adding it in a structured way. And then I let bees forage on this stuff, and I timed their visits through the array. I later caught the bee and sampled it for parasites. So this is a little bit like what it looks like. I used a database that I built on an iPad to track them between flower visits and timed every flower visit. And so you can imagine a bee entering the array, and I'm timing it. It's going through, so we know how long does it spend on each flower, how long does it take to commute to each flower. And then when I thought it was about to leave, I dropped the iPad, hopefully not in the water, and picked up the net, caught the bee, and took it to the lab the next day to analyze it for these parasites and parasitoids. We later got weather data that helped with the analysis a little because windy days were different for bees than still days, etc. And so here is a quick overview of what I collected that summer. More than 3,000 visits by individual bees, 322 individual bees of 9 species. We're only going to talk about this bee here that made maybe 90% of all of those visits. It's called Bombus vagans. Really beautiful bee that's quite common in the Northeast and other parts of the East, and up into northern Canada. There are 3 metrics that I'm interested in. So 3 measures that we're looking at. The duration of a visit. So you would think if it likes the nectar, it might stay longer. And then how far away did it fly after drinking at that flower? And how long did it take to get to its next flower? And then finally, did it fly off a flower and then come right back to it? So these revisits where the bee flies up, makes a choice and comes straight back to the same flower, which we see in nature all the time. But here I set it up experimentally. I just want to point out that in the picture on the left, this bee is she's hovering above the flower after visiting it, and she is concentrating the nectar. And you can see a little droplet of nectar on the top of the maxilla, which is the triangular shaped mouth part that holds the tongue, and the tongue is sticking out from it. That little drop is nectar, and the bees use their mouth parts to just sort of play with this stuff, and it gets some evaporation going, and then they have to store less fluid in their crops and they can forage longer. So, and you can also see pollen on that bee. So we're talking about plant chemistry, we're talking about toxins and nectar and pollen. There are those materials on the bee. I think it's important to just keep that in mind. All right, so let's look at the results. Here are the two animals, and the graphs are going to look the same for both of them, just like this. So on the left, we'll talk about how long was the visit, and on the right, we'll talk about what was the probability that they revisited the same flower. And from the post-hoc analysis of the guts of the bees, I know whether or not they had the parasite or the parasitoid, and those are symbolized by red circles and blue squares, respectively. And then on the horizontal axis there, you see the three nectar treatments. So let's see what we found. So for bees with Physocephala, the fly, there was no statistically significant effect of parasite status or the treatment. So visit duration was about the same. There was an increase in visit duration under the most bitter nectars, but it was not statistically different from the control, essentially, which would be the bees that had no parasitoid larva in their guts. And then similarly with the probability of a revisit, all three, they were about the same in both bees with the disease and bees without across the three treatment groups. They were less likely to revisit the flowers if they were especially bitter, as you can see in that graph there. Crithidia was different. So we found a significant effect of parasite status. Either it was parasitized or it was clean, on visit duration and on the probability that it would come back to the same exact flower after feeding on it. And then there's an interaction between the two factors here. One is presence absence of the disease. The other is the three treatment groups. So on the left, you can see that bees that were healthy did not have Crithidia. They ate all three nectars and they didn't seem to distinguish in terms of how much of it they took. But the bees that were parasitized, they preferred the strongest nectar. And you can clearly see that with those three red circles. They were eating a lot more of the nectar when it was especially bitter. This shows you a couple of things. One is they can taste the difference and they're doing something. They're behaving based on their sensory apparatus and what they learn about their environment, right? And then you look to the right, and there was also an effect on the likelihood that they would come back to the same flower after trying it. So the flowers that were either mean or high nectar iridoid concentration were more likely to get a revisit than the low from bees that were sick, as opposed to bees that were not sick who had a kind of the opposite pattern. So this is evidence, I think, of self-medication. These bees are able to sense what's in the nectar, and somehow they know something about their own parasite status, and they are acting on that information, both pieces of information, to structure their diet. So I thought that was really pretty amazing to see this in bees. We know that honeybees do something analogous in the hive with resins that they collect, basically that they collect resins to use as antimicrobials in the nest, but we hadn't seen this with foragers on plants producing nectar and pollen. So that was exciting. Next, we wanted to ask, how does this impact the plant? Is the plant getting anything out of being more bitter? Or maybe that's a hazard to the plants or something. So it's possible that the bitterness was attractive, but also had some detriment to the plant. Maybe it would impact its ability to get pollinated, or maybe it would help. So in this experiment, we have the same setup with flowers. They got three different nectar treatments. And then we use this fluorescent dye powder that we apply to the anthers, and it looks it's about the same size as pollen grains. And this is widely used in pollination biology as a simulation of pollen. We use it because we want to be able to find it in nature after the bee interacts with it and then goes off and forages on other flowers. So we set that up, add the nectar, add the dye, then let a bee forage on the plant, and then follow it through its next three visits. And as it did so, I carefully marked the flower in question, and then I caught the bee. Then I took the stigmas, the female parts, out of each of those three flowers. So now we know a bee has visited and gotten dusted with quote unquote pollen. And you can see the green stuff on the bee on the bottom right there. That's fluorescent dye powder on a bee that's pulling out of a flower. So she just got dusted. Now she's going to go to some other flowers, and if they're in female phase, she will be leaving that on those stigmas. And so you can see the tech here in the other photos. I think it's self-explanatory, but this was a delicate experiment and really fun to do. And in fact, we did find that the highest iridoid glycoside concentration changed things for the plant. The plant exported significantly more pollen analog dye if it was more bitter. This suggests that the plant was a better father, right? It's not that more pollen grains were coming in, it's that more of its pollen grains were going out and landing on conspecific stigmas resulting in pollination. Of course, the dye didn't result in that, but it's an analog. So this shows us that nectar chemistry can benefit the plant just as it did benefit some bees. So there's context dependency built into this for plants and for bees. And I hope you see that there's just a lot of complexity here. There's things going on in multiple directions. A plant population has a variety of different concentrations of these things. The bees have these context contingencies related to their health status. We're not even talking yet about the social biology of the animal and the fact that many of these bees are foraging for nectar to take home to feed to larvae. I didn't study this, but think about it for a minute, that this is an interesting little facet of the whole picture. And so this just shows you that some of these chemicals, while toxic to some organisms, are beneficial to bees in certain contexts. And of course, they can help plants get pollinated. So some conclusions here. There's actually a diversity of plants that make these things. And yes, they can harm pollinators. They can also benefit them. And there's often a trade-off where the pollinator gets both things on the same day. Um, the chemistry of the nectar was distinct from that of other tissues, and that shows you that the plants are behaving, the plants are doing things. It's not just a passive arrival of chemicals in the nectar because plants aren't smart enough to take care of their chemistry. Um, and then we see that the bees that are parasitized with Crithidia are self-medicating with iridoid glycosides from this one plant. We know of some other systems where this could be happening. We certainly know of other systems where the nectar benefits bees by killing off their Crithidia. And so there's more work to be done to understand whether this self-medication is widespread. We know that flowers with higher iridoid glycoside concentration have higher reproductive success. So natural selection, at least when bees are sick, should push the plants to make more bitter nectar, right? Maybe in time the parasite becomes rare, then the plants with the highest iridoid glycoside concentration might do worse. And so you can see how coevolution between pollinators and plants not only leads to speciation and all this cool stuff that we've seen through millennia behind us, but it also leads to these complex, context-dependent relationships that are often mediated by plant chemicals. So the last statement is just that plant chemicals structure multi-trophic interactions. It's not just the plant and the bee, but I've shown you it's the plant and the bee and its parasite, its parasitoid, and then also we have these herbivores that are also existing in a multi-trophic space. Trophic meaning feeding, right? A food web. So I think there are some implications of this for conservation. When we think about conserving pollinators, one of the things we do is plant flowers. And we primarily think about the carbohydrates and protein that the animals need from these flowers, that being the nectar and the pollen. Those are definitely the most important things that bees and other pollinators are getting from flowers. But we need to understand that many flowers also come with toxins slash medicines, slash behavioral manipulators. And so there may be ways we could hack some of this. For example, we may plant certain things and get a better outcome because the bees are healthier with respect to parasite infection. This is somewhat speculative, but I think it's worth trying. Um, and species recovery efforts actually, in some cases, have considered this. The U.S. Fish and Wildlife Service's efforts for the rusty patch bumblebee have incorporated some language about including plants that are beneficial in terms of this chemistry. So finally, I just want to leave you with this slide. Just remember our little plant in the middle and the bee that we care about, and just back out and look at that system. And I'm only showing you two axes of a multi-dimensional system here, right? We have a plant that's making toxins in leaves and flowers. We have pollinators that are consuming that, and then they are being attacked by parasites. The herbivores are really the same. They're feeding on foliar tissue, which definitely harms the plant, but they are doing so as to protect themselves from wasp parasitoids also. And with that, I'll say thank you for your attention, and I'd be happy to answer any questions. I want to just also say that this webinar is in concert with the podcast that Xerces produces called Bug Banter, and there's the website if you want to go find where you can download Bug Banter and listen to it. And on Tuesday next week, we have an episode coming out where I talk about some of this stuff, so it might be fun to check that out. All right, that is all I have.
Thank you so much, Leif. Like, my mind is just blown. I find this whole topic so fascinating. We're getting so many great questions in the chat, and we'll do our best to get to them all. But I thought
All
it
right.
might make sense to start back at the beginning with some of the botany and plants. So we had a question. Someone wants to know, your photo showed a common blue camas, but the text said that death camas produced
Mhm.
toxins. Do both camases produce toxins?
I am not certain. I think the answer is yes. And that was a photo that I had taken a couple of last year, and so I decided to use my own photo. We know that death camas produces toxic nectar, and I think I've read that this one does too, but I can't remember for sure. It's a good question. It's not like every plant in the same genus is equally toxic. I told you this with the milkweeds. We have an array of native milkweeds in California where I live, something like 15 to 20 species. And they have a range of cardiac glycoside concentrations in the nectar that pollinators and flower visitors in general are consuming. And so, yes, it's not all the same. So I may have I don't know, spoken out of turn with that other camas.
Thanks, Leif. All right, this next question is from Caroline or Carolyn. I am noticing that most of the studies mentioned are using toxic nectar rather than toxic pollen. I imagine that it's a bit more complex to interpret the evolutionary consequences with toxic pollen since plants incur a fitness cost with increasing pollen consumption. Can you talk about some of the benefits and or challenges to someone looking to investigate these interactions using pollen? I assume this is a fellow scientist.
That's a great question, Carolyn. Yeah, I just ignored pollen. I should have said something about it on the slide about what the potential benefits to plants are or functions in plants. For pollen typically has higher concentrations of these potentially toxic chemicals than nectar. The plant that I have talked about here, it was a little bit different, but most of them, the toxins are stronger in the pollen than the nectar, suggesting that the plants are investing more energy in protecting pollen. And as you wrote there, pollen is important to plants. It is the male gamete for sexual reproduction. So, or it contains that. And so, if pollinators ate all the pollen, then they wouldn't be able to pollinate, right? They wouldn't be able to move pollen grains to female structures, stigmas on other plants. So plants have a real interest in protecting pollen, but they also have an interest, if you think about it, in dosing out pollen on the backs of as many bees as possible. And we did actually see that with this plant where um, they can get visited many, many times and they still have a little pollen in them, and they're still sending out pollen grains on all of these many visitors. And so one thing that's interesting to think about is that the visitors to flowers that consume the most pollen are by far and away bees. Bees eat pollen because it is the only thing they feed their larvae as protein to grow the larva. Lots of other animals in nature eat animal protein, like this wasp you see in this graphic here. Bees need pollen, so of course they're on it all the time. So there's a rich place to ask questions about this stuff. How are bees interacting with pollen chemicals? You may know that can also be what we call oligolectic or pollen specialists, where they can't just eat any pollen on Earth. Some of them at the extreme end have just one plant they can get their pollen from. Um, some of them, it's one genus of plants, and there's a variety of ways that the bees can specialize like this. But if you think about it, if you could only eat one kind of pollen, um, and that pollen was toxic, you would have a problem potentially, right? A good example of this is the bees that eat the nectar and pollen of pumpkins and squashes. So the cucurbits, the genus Cucurbita, the nectar and pollen of those plants contain these really toxic chemicals, very bitter chemicals. If you've ever tasted a cucumber and the skin tastes bitter, that's that chemical. This is a related plant. And so bees, many things will not eat the pollen of those plants, but these bees that are specifically adapted to to get their food from only this plant, they seem to tolerate it just fine. So I don't, we could talk about pollen for a long time. I just want to validate your question and that there's a lot that we don't know yet about this push and pull between the pollinating mutualist, who is also a consumer of reproductive parts of the plant, right? And so this herbivory can be beneficial and it can be detrimental to plants.
Thank you very much for that. If it's not too much to ask a couple more questions related to pollen,
Yes.
would you speak more on the pollen-limited white turtle head flowers?
Yeah. So what that means is that in nature, flowers just exposed to bees for the natural amount of time are receiving visits, and each visit may or may not bring pollen to the stigma, and the plant needs a certain number of pollen grains to mature each seed. And this plant makes maybe 50 to 200 seeds in each fruit. And so what we can do is just take the fruits and count the number of seeds. And then experimentally, we can add pollen to other flowers, wait until they become fruits, then take those fruits and count the seeds. And we find more seeds in the ones that we add pollen to. So what this tells us is that in a natural situation, not enough pollen is from the right plants is coming in to this plant's stigmas. It could be for a number of reasons. It can be because in some plants they are self-incompatible. That's not the case with this plant. But some plants, they cannot self-pollinate. And if a lot of their own pollen gets onto their female parts, then pollens from outside can't even reach the stigma. So we call it gunking in the business. Sometimes that can happen. Um, Basically, it just means that the plants are not optimizing their reproduction. And I chose to include that graph because I think it shows you that the plants, in an evolutionary sense, in an ecological sense, they have a vested interest in getting more visits. So they should be trying to attract floral visitors if they are trying, quote unquote, to maximize their reproduction, which is kind of an assumption of plant reproduction. So that's pollen limitation, yeah.
All right, next question from Sego. If it's not too far off topic, can you speak to how common mass bee mortality is with linden trees, which is interesting because we talk about this in the podcast. After some quick Googling, we decided to not plant linden trees because of this
Uh,
potential. Was that a good decision?
oh, this is complicated, and I don't have a satisfying answer. For a long time, people have observed that some linden trees are toxic to some bees. This particularly started in Britain, where people were noticing bumblebees and honeybees, especially the latter, being killed by foraging on these plants. And this is another one of these plants where they fall off the plant, and you find dead bees around the base of the plant. Um, There's a ton of literature on this where people try to explain it. And I think we've arrived at the best answer right now is that there is a compound in the nectar that's toxic to some consumers, and it's actually a sugar. I think it's mannose, one of these sort of exotic sugars that bees can't digest, apparently. So that's one explanation for that. I've seen other explanations. It could be some other chemical we're not finding. It does sound like some lindens, some trees in the genus Tilia are not toxic and others are. And I should hasten to add also that we know that in some cases we see a lot of dead bees under linden trees because of pesticide applications to them. There's a kind of famous case, notorious case from Oregon that some Xerces folks worked on and published on where roughly 75,000 bumblebees were killed by foraging on a parking lot's worth of these trees because the trees had been treated with a systemic insecticide that was being expressed in the nectar. So um, some of the worry about linden could come from things like that as well. But yeah, it's probably a sugar or some other chemical. And as to whether or not you should plant them, I don't know. I feel like these things are naturally occurring, and this is part of the buffet that bees and butterflies and wasps and ants come to. And in my own garden, I plant lots of toxic things because I like plants. I like some of these cool exotic toxins. And, they might not be great for all of the bees, but I think it's important to understand, the parasitoid wasp on this slide, not a bad thing. It is just a part of the natural system. And so is the chemistry there. So if you love that plant, as I do, and I wasn't going to bring this up. My daughter's middle name is Tilia. I love those plants. I would plant it just because it's beautiful, smells great, and has amazing nectar properties. So that's how I feel about the garden question that you asked.
I love that, Leif. Thank you for sharing that little detail. And yeah, in my own garden, when you said that the turtle head is related to monkey flower, I have a lot of monkey flower, and I'm gonna go look at those a little differently and inspect them a little
Cool.
closer. All right, another question from a listener or a viewer. Any early information on generalists versus specialist bees for this plant usage of chemicals?
Oh, that's a cool question. So I don't know too much about it. I'll say some of the specialist bees that are restricted in their diet of pollens are exposed to some pretty gnarly chemicals, and so we would predict that they can tolerate them or they have some immunity to them. Um, Specialists are not always the best pollinators for the plant, but they tend to be pretty good pollinators. There tend to be other visitors to the flowers as well. So you might have the specialist with some co-evolved adaptation to tolerate the toxins in the pollen, whereas the generalist flower visitors cannot eat those same chemicals. And so maybe the plant is kind of filtering the visitor spectrum. So it gets more pollinators and fewer of these robbers or thieves. Um, I'll say also a question people have asked is, why are these female bees restricting their diet? There's all this good stuff out there. Why are they only taking pollen from this one thing or this one thing that is even a little bit toxic? And there's one experiment I can think of where people fed the wrong pollen to larval bees that are specialists or oligolectic. And in that study, they found that the larva could develop just fine on pollens that had different chemistry from the one their mom would have chosen. So I don't think that that means necessarily that the moms don't know what they're doing, but it does suggest that at least the larva can consume and digest other things than what it's being fed. And so I think there is actually a lot more interesting questions to ask in these specialized systems where a bee is, or well, a bee is wholly dependent on a restricted set of plants. The chemistry of the plants should matter more, right? It doesn't have options to do something else.
All right, this is kind of a follow-up from that, maybe adjacent a little bit. This is from Dave. Has anyone looked at whether toxic pollen may provide protection against kleptoparasites in the case of specialist bees able to tolerate the toxin pollen?
Oh, that's a great question. So I think the answer is no, but it's a very good question. So kleptoparasites are bees that are parasitic on other bees. And when the host mother is out of the nest foraging, the kleptoparasite comes in and lays an egg and then leaves quickly. Klepto means to steal. And in this case, I guess they're stealing the resources, but they're also stealing in and laying an egg. They often hide the egg under the pollen provision in the nest or in the wall of the nest so the mother isn't aware of it. After the mother closes up that cell, the kleptoparasite larva outcompetes its host, eats all the pollen, maybe eats the host and emerges from the cell. So if the mother provisioned the cell with some toxic pollen, maybe that would protect that bee from its kleptoparasite. It's a great question, and I'm not aware of any research on this yet. But one prediction that might come from that question is that bees with more specialized diets, sorry, bees that have more kleptoparasites should be eating more toxic pollens. And that's a question we could answer with existing data, probably just looking at patterns of flower visitation and what we know about these host parasite interactions. So again, I don't know about research in that area. I'm sure other people are thinking about it, and I hope someone else looks into that because I think it's a worthwhile set of questions.
Yes, I agree. There's just so much more to learn here. It's fascinating. All right, another question. Is the presence of toxic substances in flowers changing with the seasons?
Ah, um, yeah, to some extent. So yes, the quality of floral nectar, I'll say, I don't know about pollen, but pollen is produced and it doesn't really change. It just sits there. It dries out slowly. So maybe the chemistry changes a little bit. Nectar chemistry does change quite a bit. For example, on the first day the plant makes nectar, it has a certain concentration of sugars and other chemicals. Um, Nectars often dehydrate in the hot, sunny days and such. You may then find a higher sugar concentration in that same nectar. And I would expect that the plant chemicals would be more concentrated as well, these toxins or potential toxins. There are nectars that get attacked by yeasts. These are really cool yeasts. One of them is called Meshnicovia. It's a specialist on bees in that we think it probably lives in the bodies of bees, and bees infect the nectar with this Meshnicovia thing, and it ferments the nectar. So we can't find it in the winter when flowers aren't around. We find it in nectar of flowers and on bees, I guess. And so the idea is that bees are distributing it to its specialized habitat, which is inside of flowers where there's nectar. Some of these things, I can't remember if this one in particular does this, but some of these microbes ferment the nectar, and then in the life of that flower, you get alcoholic nectar. And there's a really cool study that shows that bumblebees are attracted to that alcohol, and they're more likely to visit flowers that have alcoholic nectar than flowers that don't. So there's an example of nectar changing over the short lifespan of a flower, maybe 1 to 5 days or something. And I think it's possible that the plants would change their expression of chemicals in nectar over the course of a season or something based on, responding to the environment. And the last thing I'll say about that is that nectar secretion dynamics are really complex and fascinating. It reveals all sorts of cool stuff about plant life. The time of day that they secrete, the question of whether they resecrete nectar if a pollinator or a visitor takes the nectar away. Some do, some don't. And then there's this cool thing where plants can resorb nectar. So they're saving the sugars that are not consumed by pollinators. I would bet you that they're also saving some of these chemicals. So they are literally pulling the sugars back into the plant out of the flower, maybe at the end of the flowering period or something like that. Maybe after they get pollinated, they pull back the reward. I'm just generalizing here. But yes, nectar, the quality of nectar can change a lot in the short lifetime of these flowers, and maybe over the course of a longer time scale, a plant's flowering period or something. Um, I just want to add that we know that other organisms can affect nectar chemistry. In fact, foliar herbivores can affect nectar chemistry expression. So some plants, when they get chewed on the leaves, the nectar chemistry changes. I did some other research where we found that beneficial mutualist fungi that interact with the roots of blueberry plants change the quality of the nectar of the plants and make the plants more attractive to pollinators. And so nectar is changing in all kinds of ways. And again, just like the system I'm showing in my little graphic here, there's all this context dependency and ecological context that we have to think about. So yeah, a bee arrives at a flower and she's going to use her sensory apparatus to figure out what's going on. What's the sweetness? What's the bitterness? Do I want to take this home or not? And there are many different things going on at once, in my view.
It's so interesting. So this is a similar question. Carol's wondering, how would drought affect the toxicity of some of these chemicals? Is there a dilution effect during rains?
Yeah, if the flower's open to rain, yes, the nectar would be diluted by raindrops falling into it. Um, there are Plants do change their chemistry during drought. There's a wide array of things that plants do with chemicals. One of them, some chemicals help plants deal with drought stress. And so they may express more of those chemicals in dry environments or in dry seasons than in wet seasons. There's a ton of research on drought showing that drought affects flowers and the resources that pollinators are withdrawing from them. So that could certainly affect the nectar, and not only in obvious ways like just reducing the volume of the nectar or rain increasing the volume and decreasing the sweetness of the nectar. Plants are responding to their environment, and manufacturing of chemicals is a dynamic process. Plants can upregulate certain genes and express certain things, and then downregulate them and stop expressing them. And you've hit something that's really true. Drought does affect the quality of flowers and their rewards.
Thanks for that answer. Yeah, getting so many questions. We'll keep trying to get through them. Um, Going back to your specific study here for a minute, in the study with the 8 chemicals and the controls, how did you differentiate the action among the 8 chemicals on the parasite?
We didn't. We did not look into mechanism. So how are these chemicals resulting in fewer parasites in the gut after 7 days? We didn't ask that question. It's possible. It's a great question, and I've speculated about it in some of the writing that I've done about this. It's possible that the chemicals are toxic to one or more life stages of this parasite, it just kills them. It's possible that it changes the gut lining in some way and makes it harder for the parasite. These parasites suck they live on food in the gut, but they also attach to the gut lining, and I think they can even suck hemolymph through the gut. And these chemicals might protect the lining of the gut from the things attaching. One way that bees cope with gut parasites is pooping. They just flush it out, and so you could imagine that the ecology of the gut could change in the presence of some of these chemicals. Maybe it's harder to hold on. There are fewer places to hide, or something like that. So we did not look into this, but there are some nice hypotheses about how this may actually happen. I mean, in general, I would just say that these chemicals are probably toxic to those microbes. And I also just want to point out, you might have caught this in the experimental design, but I inoculated all the bees with the same number of parasite cells. It was a low dose, and then the parasite infection grew over 7 days. So really, when I say there are fewer parasites in bees that took nicotine than bees that didn't take anything, um, it really means that they grew more slowly under the influence of nicotine. It isn't like they had a high parasite load and then I knocked it back with a chemical. It's the onset of the disease that we experimented in that experiment.
Right. Another specific question about one of your studies. Cedar is wondering, your graph of visit time for the Cryphidia infected bees showed significantly lower visit time on the less bitter turtlehead flowers compared to the uninfected bees. The
Yeah.
higher visit time for more bitter flowers among infected bees was the same as uninfected, not
Yeah.
more. What do you make of this?
Right. So the uninfected bees just did not make a distinction between the three types of nectars in terms of how long they sucked on them. And that duration of the visit is a proxy for how much nectar they're consuming. It's also a proxy for how much pollen is being deposited on their bodies as they forage for that nectar. Um, so what I make of that graph is just that when the bees are healthy, they're not avoiding iridoid glycosides. That's actually important, right? That it doesn't seem like, even though they can taste this bitterness, they're not going out of their way to avoid it when they're well. They're still consuming it. Maybe we could explain it with a just so story, like maybe some of the individuals in the nest are sick and they are bringing it back to the nest, whether or not they themselves are sick. Um, but they're not foraging specifically for those chemicals based on that graph, that is what I would say. Whereas parasitized bees showed a clear difference in behavior, in duration of consuming nectar based on its chemistry. Yeah.
All right, thanks for that. Back to a subject you brought up a couple questions ago. Someone has seen many times bumblebees seemingly intoxicated and unable to fly out from cardoon flowers. They've always assumed
Huh.
that they were nectar drunk. Might something else be going on here? The bees are often still on the flowers in the morning, but seem to head off eventually.
Uh, I can't say what's going on there. I'm not sure offhand if cardoon contains nasty chemicals or not. I think that's a thistle. It's possible there's something going on there. Um, yeah, the bees could be inebriated by some psychotropic drug that they're getting out of the plant. They could be literally drunk on the alcohol that I just described. And then unfortunately, when questions like this arise, we often get to the root of the cause, and it turns out to be pesticide exposure. So, bees that act drunk or lay on their backs and wiggle their legs or twitch, um, we can't say what happened, but those are classic symptoms of pesticide exposure. And so sometimes it's not plant derived chemicals, it's human derived chemicals. So I'm just speculating here about your situation and your question, but um, all of those things are probably possible. Yeah.
All right, I have a couple questions I'm going to combine here, just specifically about, and we may not know the answer to this, but do bees know the quality of nectar and pollen before they land on the plant? Do they know what the toxicity level is?
Yes and no. In this system, there's a lot of evidence that they can taste it. They're making decisions based on the concentration of the nectar that I put in the flowers. I'm pretty certain they can taste it. I'll just add, I can taste it. These iridoid glycosides are extremely bitter. They've been described as some of the most bitter compounds in nature. Um, and I personally like the taste of olives quite a bit, but it's that bitterness, that astringency that is one of the nice things about olives. So with these chemicals, yes, we think that they can taste them and make choices based on that. There are certainly some chemicals they cannot taste, and there you wonder, maybe they're not making decisions so much about consuming these things, but consuming them is incidental to consuming the carbohydrates and protein that they need from the flowers. And it may have some biological effect on them, even though they aren't self-medicating, as I showed in this talk.
Thanks. Okay, we have time for just a few more questions here. Claudia would like to know, can you recommend a white turtlehead pollinator observation protocol for them to try on their own? In other
Oh.
words, they want to sit and watch the turtleheads in their rain garden. How best to watch for this pollinator plant interaction? They're seeing the most flowers they ever had. They had a super wet early summer, so maybe that contributed to the increase this year.
Well, first I'll say you're lucky to have this plant in your garden, I think. There's another one people plant in the East, that's called Hot Lips, and it's got bright pink flowers. It's really common in gardens in New England. That one, I tested it. That one has glycosides in the nectar as well. There are only a few species of this thing, maybe 4 or 5, and they're all in Central to Eastern North America. And so some observation protocols. I mean, the simplest thing to do, something I do all the time when I'm in the field, is just sit down and watch. Maybe you have a notebook with you. Maybe your question is how many bees visit per minute, or how many seconds are they spending in a flower? How long does it take them to pry open the flower? I didn't say this earlier, but this plant has a largely closed corolla, and the bees have to learn how to crack it open and get inside to get the nectar and pollen. And naive bees can't do it very well. It's kind of hilarious to watch. They fall off the flower. They're using two of their forelegs to pull on the thing. Once they get it, though, it's almost like they're just flying right through a wall. They just slip right into the flower in a fraction of a second. Maybe that's interesting to you. And so you might record something about the behavior of those bees. Um, Surveys of this sort are often effort limited. So that means they're limited in their time or some other scope, so that we know we can compare your survey to your survey tomorrow and my survey today, and so on. So you might think about doing a standardized amount of time. Like, I'm going to sit in my garden for 15 minutes and watch every visitor, and I'm going to record the the visitors to whatever extent I can, and then maybe I'll do it at a different time another day, and see what's happening. I'll say also with this plant, you should always be careful around bees. You can get stung, and stinging is painful and sometimes is very dangerous for us if we're allergic to bee stings. But this plant, the bees go inside the flower and they cannot see you. And so you can sneak right up on them. You can get amazing photos of bees coming out of turtlehead flowers. They're completely unaware that you're there, and you can be an inch away from them with the camera. It's a great place to do some wildlife photography. So if your interest is more in just immersing yourself in the organisms in the story, the safari of it all, I encourage you to use this plant as a place to take photos. It's a really good one to start with.
I love that. And I love that question. All right, Dylan has a question related to seasonal toxins and droughts. Noticing that plants during the summer out west have a lot of oils and chemicals externally, I assume pesticides, and possibly trying to use some oils instead of pesticides. Do these external chemicals influence flower visitors at all?
Absolutely. I didn't even mention it, but flowers smell great, right? Those smells are plant secondary metabolites. They're just very small molecules that volatilize easily. So the plant is producing, actually on the corolla tissue, not in the nectary, but right out there on the white, in this case, whitish pinkish tissue. They're producing chemicals in these tiny little glands that are just volatilizing right off of the plant into the air. And that's the way the bees and the other organisms are finding the flowers, right? So, yeah, there are a lot of other chemicals besides those in just nectar and pollen that affect visitation. Um, some plants have hairs of various sorts that contain chemicals. Uh, Penstemons are actually, some of them are classic examples where there are these little trichomes that have nasty stuff in them, and they may deter some herbivory, but they smell and they're sticky. And pollinators often land on the inflorescence or the stem or something before they go to the flower, and they are certainly interacting with those things. And so I think in general, we can speculate that, yeah, chemicals in other parts of the plants are detectable by these animals in some cases, and that they may make choices and behave as a consequence of them. Um, uh, there are also studies that show that there are these multi-trophic interactions where, like, the chemistry of the leaves and the stems is important to keeping ants from doing something to the plant. And when the ants are present, the bees are behaving differently on the flowers. So sometimes there's an indirect effect of foliar chemistry on the flower visitors that's mediated through other animals.
Thanks for that, Leif. All right, we do have to cut it off with just this one last question here. We have so many more. If you'd like, please email outreach@xerces.org. We can try and get some questions answered. We're sorry we can't get to them all. Last one here for you, Leif. Another layer here. Any attempt to extract paleotoxins from plant remnants, including pollen from amber or lacustrine, lake or glacial deposits, to document any possible timeline for this coevolution?
That's a cool question. I am not aware of any research like that. Um, Generally, I think these chemicals are going to be long gone by the time something fossilizes. They're in the case of the nectar, it's in solution. It's molecules that probably aren't detectable after long periods of time. But on the other hand, archaeology has made great strides in analogous analyses where, we know someone was brewing beer in Egypt 6,000 years ago or something, or the seeds that were ground in this little bowl are the following three things, and that kind of stuff. So it's possible that there's some sort of paleo chemistry that could be done there. And then um, we're able to sequence the genomes of some of these fossilized extinct things. And if we could identify the genes responsible for the chemicals, then we could look for them in these older plants. And I would guess that a person interested in your question would start there, would start with the genetics and look for changes over time in the genes that are thought to have something to do with the expression of these chemicals.
Thank you so much, Leif. Thank you for answering all these questions and just exploring this really interesting topic. I feel like this whole new world has been opened up to me after doing the podcast and then learning even more today. So thank you so much for your time, and thank you to the audience for all the wonderful, thoughtful, really ingenious questions. We hope to have you back next month for our next webinar, and check out Bug Banter, the next episode with Leif, coming out on this following Tuesday. And yeah, thank you so much for your time. Thank you, Amber, as well, for co-hosting with me. I hope you all have a wonderful day. Thank you so much.
Thanks, everybody.